Hello everyone. In this video we want to talk about the function and the DNA. We studied the structure of the DNA and it's made up of what. Now we want to talk about the things the DNA is doing in the cell. The first thing we want to talk about the copying of the DNA, or something, or a new term or process which is called replication. When we studied, if you remember, when we studied the meiosis and mitosis for the cell division, we said at the beginning, in the interphase, all the DNA will be copied. So the DNA, to give the correct number of the DNA, so it should be copied, it should be replicated. Replicated, it means what? It makes what? Copies of the DNA. So the cell makes copies of DNA molecules through a process known as replication. In this lesson, we want to study this process of replication. What is replication? Okay, so how is replication? How the DNA will be copied? During replication, actually, if you remember, we said for the DNA it's double strand, right? And the DNA, if you remember also, we have mentioned, I want you to look at this karyotype. If you remember, we said each chromosome, it will be copied. Yeah, this one will be copied. This one will be copied. Then they will make the X structure, right? So how it will be copied? How they will make the copy of this one and copy of this one? This is what we want to explain for today. To understand this, let's go back to the structure of the DNA. As you know, the DNA, it's a twisted double helix, which is a double helix structure, and you know it's made up of double strands, and these double strands, the backbone of this double strand, which is a P phosphate group and sugar, and here in the middle the nitrogen bases, they are connected together in hydrogen bond. For the DNA to be copied, the first step that will happen to duplicate or to copy the DNA, the first step it happened during replication, these two strands of DNA separate. So the first step, two strands of the DNA, you have to write here number 1. The first step to make replication or to copy the DNA, the two strands will be copied, it will be separated. Okay, then the bases on each side of molecule are used as pattern for a new strand type. What does that mean? Let's study this. This is the second step. The second step, the bases on each side of the molecules are used as pattern or a new strand. To understand this, I want you to look at this first. We said for the double strand will be separated from each other. It's on separated, and you know, if you look at this strand when it's open, okay, you have to look at the nitrogen bases here. And these nitrogen bases here, they are gonna be as what? The pattern. Yani if this one here is C, it will I have to add what? G. And the same one here, if this one is G, I have to add here what? A C. Let's understand this by making this one. Look at this one. Imagine you have a DNA strand. This is DNA strand. I'll do it with you. I'll take a segment, not all the DNA for sure. I make it. You have to make it. It should be twisted for. And as you know, this is the two strands, and these two strands they have here. Let's write the bases. For example, here you have A. It should be connected with what? It should be connected with T. Maybe in the exam I'll ask you to fill what is the missing. Okay, that here imagine I have another A, also it's connected with T. That this one is C, it should be connected with G from the other side. G, not C, which is wrong. That this one here, G, it should be connected with C from the other side. These strands, they are complementary. Complementary, they complete each other. Look how I'm doing this. This one here is T. It should be connected with A. You have another, for example, A. It should be connected. They are complementary to each other, okay? This one is T. This one is C. This one is G. Let's say another G or another C with a G. This is a DNA now, imagine. And this DNA, I want to copy this DNA. This is strand number 1, I'll call it. And here, this is strand number 2. The first step to replication, to make the replication process, this DNA, these T with A, they are connected with a hydrogen bond between them. They are connected together in a hydrogen bond. The first step, these two strands should be separated. So I'm going to put a red color here. This one, it should be separated. So how to start with the first one? This is 1. I'm going to put it here. I'll take the first strand, this one, okay? I'll take this one. I'll take this one, I'll make a copy of it. Hello, what's gonna happen? This is the first strand. And I'll put these are the you have here A, and this one C, this one G, this one T, then you have A, then you have C and C, right? So they should be correct, the same. 1 2 3 4 5 6 7 8 1 2 3 4 5 6 7 8. Okay, so this is for the old strand. This is the old strand, right? This is the old strand, and which is number 1, right? For the nitrogen bases, they're going to be as what? As the as a pattern. يعني start to build now a new strand. Now I'm going to build the new strand. I'm going to use a different color to show you the new strand. This is a new strand, and it should be complementary to the old strand. So I'm going to add them. A, I have to add what? T. And A, I have to add here T. And C, I have to add here G. And G, I have to add here C. With T, I have to add here A. With A, I have to add T, C, G, and the other one is G. Now, this one here, as you see, this is the new strand. Let's continue. I didn't finish yet. I still have this old strand number 2. Strand number 2, I'll put it here. I'm going to use this color here. So this one here, the old strand number 2. The old strand number 2, let's say. It should be what? I have to put the pattern of it, which is the sorry for this. Let's continue. Here you have T, I'm going to copy them from here. This is strand T, the new. This is one, which is the old, sorry. This is the old, okay? T, you have another T, and G, you have C here, you have A, you have T, you have G and G, right? G and G. The new strand is formed depending on the old one, which is because the old one, it's like a pattern of it. It should be like this. This is the new strand. This is the old here. I'm going to write here, this is the old. And here, this is the new with the blue one. So I have to write the complementary. This is 1A. This one A, this one G, sorry, C, this one C, because we have G, and this one G, this one T, this one A, this one C, and this one is C. So, as you see here, and this is the new strand is built. Sorry. So here, as you see, we have two copies. This is the copy number 1. Copy here, this is 1 and 2. You have now 2 instead of 1. So I have a copy of it. This is the replication process. Okay, this is the replication process. So if I want to talk about the steps of replication, let's go to the book to talk about the steps of replication. Actually, let's go talk about them in more details here. The first step, what happened in A, let's say here, the DNA molecule, I'm going to write here, the DNA molecule opens up. The DNA molecule opens up. It should be open. The double strand should be open, okay? Opens up, separating into what? 2 strands. It will be separating into 2 strands. This is the first step to make the replication, okay? The second step, what happened? For the second step, nucleotides, here I'm going to write here, match up and join. This one is a nucleotide. Nucleotides actually here. Let's talk about this one. The nucleotide. What happened to the nucleotide? Just to make it smaller. So, a nucleotide match up and join to the open DNA strand. Yani, I open this one, a new nucleotide will be added and match to the old one, okay? Yani, A with T, T with A, C with G, G with C, okay? So, then, what's gonna happen? The last step, in the last step, the 2 identical molecules are formed. 2 identical molecules are formed. Okay, so here for the DNA replication, the first thing, the DNA should be open into 2 strands, then a nucleotide will be added to the match, should be joined and match with the old strand, okay, and they should be complementary. Then identical molecules are formed at the end, okay? So when replication is complete, there are 2 identical DNA molecules. Each new DNA molecule is made of 1 strand of old DNA and a strand with a new. If you remember, let's go back to this one. One of them, all the other one new. This one is, here is this one here is a new, and this one is old. Okay, when copies of DNA made this replication when? Before a cell divide, it copies the DNA so that each new daughter cell will have complete set of structure. So our cells can replicate DNA in just a few hours, and it's very speed process. Okay, replication actually begins in many places along DNA strand. So many groups of proteins are working to replicate your DNA at the same time. يعني this process, it's not only opening like this. Many different proteins and enzymes are working to complete this process. I'm going to show you now a video to help you to understand. Actually, the name of these enzymes are not required from you, but I want you to يعني be familiar with these protein enzymes. Okay, listen. For this now, it's enough for now. Next time I'll show you a video about the replication process. If you have any question, you can send me your question. Thank you for listening.